सो सॉरी स्टूडेंट, पिछली वाली वीडियो क्लोज हो गई थी। सो हम लोग डिस्कस कर रहे थे दैट वी आर डिस्कसिंग द फिजिकल मेथड। सो इन द फिजिकल मेथड, द मेथड इन विच you change the physical conditions of the environment to kill all of these microbes. Okay, so this is called the physical method. Now let's discuss what are the types of them. So there are two types: cold sterilization and heat sterilization. Then the heat sterilization is again divided. So first we are going to see this heat sterilization process. So let's start it. So here the heat sterilization method is given that the microbes are killed by heating process. Okay, so various microbes are present which cannot bear the high temperature, so they killed by it. And due to the heat, their protein and their enzymes become coagulated. So therefore, this process is used. It consists of two types: moist heat and dry heat. So first, let's discuss about the moist sterilization. So, moist sterilization is a technique in which hot air is laden heavily with the water vapor. So, there is a presence of water vapors in your air. If your steam is highly consists of water vapors, then this is called as moist heat sterilization. So, there is very important role of water vapors present in the steam because they can help in the prevention of growth of microorganisms. So there are again three divided methods of sterilization. Like the first is the moist heat below the temperature of 100 degrees centigrade. Then we can do the moist heat sterilization at 100 degrees centigrade, or we can do moist heat sterilization above the temperature of 100 degrees centigrade. So there processes are also given, like below 100 the pasteurization process is done, at the 100 temperature the boiling water and the tendilation effects are observed, and above the 100 the process is used which is called as autoclave. So there are bacteria which can killed at the temperature below 100. There are some bacteria who need above the temperature above than the 100 degrees centigrade, so they are placed in autoclave in order to kill them. Now let's discuss all of these four processes. First is the pasteurization. So like a bacteria which is called as salmonella, it is present in the milk. So it tributes the milk borne diseases, the diseases which are caused due to milk. So in the milk actually this bacteria is causing the diseases. So what we do, we will pasteurize that milk in order to kill this particular bacteria which is present in the milk. So how you will perform in, so in the pasteurization you will kill your this bacteria at 63 degrees centigrade mean temperature for the 30 minutes. So you know that the temperature is below than the 100, and we have said that the moisture below 100. So at the 63 temperature this bacteria will break down or removed or killed, and then this milk is purified and it is ready to drink. And other method is that you increase the temperature but decrease there in the time. So first is for the 30 minutes. Now the increasing the temperature towards the 72. Now just only you need 15 to 20 seconds, and after that your milk is ready to go. Second method is the boiling method. You know that the boiling water is very helpful for the removal of all types of bacteria. Like you have to heat your water at the 100 degrees centigrade, and then you can add 2% sodium carbonate in it, and by adding 2% sodium carbonate it increase the disinfecting power of your boiling water. And after that you will put something that you want to clarify, like you can put surgical instrument, you can put glasswares in it after that, in order to clean them or disinfect them from all types of microbes. Then third step is sterilization, which is also used at the 100 temperature. The steam, which is at 100 temperature, it is used to kill all the microorganisms which are present on the surgical instruments. So surgical instruments should be sterile before the use. So in order to, there is no transfer of microbes in the patient body. So therefore they should be sterilized, and we use these processes to sterilize them. And the glassware which are used in the laboratory should be sterilized, and they are sterilized by this process. Now we have discussed pasteurization, boiling water, tenderization. Now the last process is the autoclave. Now let's discuss it. So here the autoclave is given. That autoclave is also called as a pressure cooker, which you use in your daily household. So it is just similar to a pressure cooker. So first of all, it was discovered by a scientist which is called as Charles Camberland. Definition is what that it is an instrument which you use to sterilize equipments at the temperature of 121 degrees centigrade for 15 to 20 minutes. So now the temperature is above the 100 degrees centigrade. In the autoclave, pressurized steam is used to kill the bacteria. So in this autoclave and in the pressure cooker, you know that the steam is going to pressurized in it, and this pressurized steam is very helpful to remove all kind of bacteria. So we will increase the pressure inside the pressure cooker or inside this autoclave, and we will decrease the bacteria. What is the mechanism of action? How the pressurized steam can kill all the bacteria? Actually, this pressurized steam cause the coagulation or denaturation of bacterial proteins. So all the types of, you know that every organism is made up of protein, made up of amino acids. So when the temperature exerts on them, there is the coagulation or denaturation of their proteins, denaturation of their enzymes. So you know that no one can survive without these protein and without these enzymes. So therefore these bacteria will kill down. Now the next is the dry heat sterilization. So in the previous we have discussed what is the moist heat, we have discussed what are the processes of them, we have covered all. Now it is time for the dry heat method. So in this technique your hot air is free from water vapors. Now there is no water vapors in it. It is totally free from it, but the moist heat contain water vapors, is doesn't contain. Again, it is divided into three processes: hot air oven, incineration, and direct flame. First, discuss the direct flame. So, you will place your instrument which you want to be sterilized, like surgical instrument. You will directly place over them over a Bunsen burner. So, you know, Bunsen burner is an agent which release the heat out of it, and it is using in heating various processes of it. So therefore, in the direct flame, you will place over the Bunsen burner. After that, it is ready to use. Second is the hot air oven. Now in that hot air oven, you will use the temperature of 160 to 180 degrees centigrade for 1 hour. Okay, so then this oven, you will place your instrument in this oven. Temperature is this for after 1 hour. Your instruments are sterilized. Last process is the incineration. Incineration is something else without him. It is the electrical heat air inside it. So actually, what is the incineration? What are the apparatus of it? So these apparatus is consist of this shape, as you can see here. Okay. So now there is a spring on its wall, and this spring is electrically connected, mean it is connected with the electricity. Okay, so one end connect with the one end of the electricity, other end connects of one end. So this is the positive, like this is the negative charge is given. Now the electricity flow in this spring. Now it is closed from the upper end. Now the air is present inside this apparatus. Okay, now what is happen as the spring is heated up, because the electricity pass in this spring, so it can also cause the heating of the air inside it. Okay, now this is the process in which electrically we heat the air inside it. It used to destroy the solid dressing. एक्चुअली ये क्या है, कंबशन प्रोसेस है जिसमें आप ऑर्गेनिक सब्सटेंसेस को रखते हैं जिनके अंदर वेस्ट मटेरियल होता है। लाइक कोई भी मैं ऑर्गेनिक सब्सटेंस, ये एक बहुत बड़ा ऑर्गेनिक सब्सटेंस है, मैंने इसको इसके अंदर रखा है और इसके अंदर वेस्ट मटेरियल्स हैं, मैं उनके कंबशन चाहती हूँ। और कंबशन से आपको फॉसल फ्यूल्स मिलते हैं, आपको एनर्जी मिलती है, उसी एनर्जी से हम अपने जो है वो डैम्स चला रहे हैं, हम अपनी इलेक्ट्रिसिटी उससे जो है वो प्रोड्यूस कर रहे होते हैं। सो डिफरेंट चीज़ें हम लोग जो है उससे यूज़ कर रहे होते हैं। सो इंसलेशन में यही चीज़ होती है। अब आपने सर्जिकल में अगर मतलब मेडिकल में इसका देखें तो डिफरेंट किस्म के जो सॉलिड ड्रेसिंग्स होती हैं, जो पट्टी करने के लिए जो भी चीज़ें यूज़ होती हैं, या और भी कोई चीज़ जो हमारी वेस्ट हो चुकी है, अब उसी वेस्ट से हम एनर्जी प्रोड्यूस कर रहे हैं कि हम उसी जैसे सॉलिड ड्रेसिंग्स हैं, हम उसी को कंबस्ट करेंगे, उसके अंदर जो ऑर्गेनिक सब्सटेंस हैं जो हमारे लिए एक वेस्ट मटेरियल है, वो कंबस्ट होगा, वो जलेगा और उसके जलने से हमें इलेक्ट्रिसिटी मिलेगी, हमें एनर्जी मिलेगी। सो ये प्रोसेस यूज़ हो रहे हैं हमारे ड्राई हीट स्टेरिलाइजेशन के अंदर। और इसी के अंदर आप लोग क्या करेंगे, कोई भी अपना सर्जिकल इंस्ट्रूमेंट यहाँ पर प्लेस कर सकते हैं। और जो स्टीम है वो उसको वार्म अप करेगी, उसके अंदर सारे बैक्टीरियाज जो हैं वो मर जाएंगे और फिर आप अब उसको यूज़ कर सकते हैं। सो ये था हमारा पहला प्रोसेस, जिसमें हम लोगों ने देखा फिज़िकल मेथड के ज़रिए से हम किस तरह स्टेरिलाइजेशन कर रहे हैं। फिज़िकल मेथड में भी हमने हीट स्टेरिलाइजेशन के जो 2 टाइप्स थे, ड्राई हीट एंड मॉइस्ट हीट स्टेरिलाइजेशन, उन दोनों को डिस्कस कर लिया हमने। नेक्स्ट लेक्चर में फिर हम लोग कोल्ड स्टेरिलाइजेशन देखेंगे, रेडिएशन के ज़रिए से किस तरह से हो रही है, और उसके बाद हम मैकेनिकल मेथड ऑफ फिल्ट्रेशन पे जाएंगे। सो आई होप कि आप लोगों को आज का ये लेक्चर अच्छा लगा होगा। अगर आपको वीडियो अच्छी लगी है तो प्लीज़ वीडियो को लाइक कर दें, चैनल को सब्सक्राइब कर दें। मिलेंगे नेक्स्ट लेक्चर में नेक्स्ट टॉपिक के साथ, लेकिन तब तक अपना बहुत सारा ख्याल रखिएगा। अल्लाह हाफिज़।